Dr. Carter, how good to have you with us today.
Thank you.
Now we're going to talk about Eve today. Is she the mother of all mankind?
According to the Bible, she should be, and according to a lot of evidence in science, she is also.
So you think that what the Bible says and what the evidence of science says, it brings you to the same conclusion?
Yes.
Okay, well help us understand that.
Okay, well before we get into that, we need to talk about just one particular thing in genetics.
Okay.
And that is, what is a gene? What is a piece of DNA? What is a genome? This is a picture of a person's genome.
Okay.
There's 3 billion letters amongst 46 molecules. Our molecules are called chromosomes. And they're broken up into these pieces, and the things that are written into those letters there control everything about us. This is the part of the genome that's inside the nucleus, that's inside our cells.
Okay.
The part that we need to focus on to talk about Eve is a piece of DNA that's only inherited from your mother. But it's not in the nucleus of the cell. It's actually outside in a little organelle called the mitochondrion. And the mitochondrial DNA controls the conversion of sugars into energy. And so you have in each cell you have about 100 little mitochondria, and they're little energy factories producing energy for us to live and breathe and think and things like that. And according to theory, and according to a lot of experimental evidence, you only get that from your mother. And it's critical because that means that what we can do is we can take all the mitochondrial DNA in the world and look at them, compare them, and then build a family tree of all the women. And that family tree points back to a single female.
Wow.
But that little piece of DNA has been used to develop what's called the out of Africa theory.
Okay.
The evolutionists came up with this term. It's called mitochondrial Eve. It's the female ancestor who carried that. And they get it from this particular chart. This is a chart from the original paper that came out in 1987.
Okay.
It's very complicated looking, but if you look on the very bottom, on the right, there's a group of sequences there. They only come from Africa. And they're very different from the sequences in the rest of the world. In fact, here, I'll circle the African sequences.
Okay. Those are only in Africa?
Those are only in Africa. But there are also some sequences from Africa that are found that are similar to the other ones also.
All right, got it.
Now, the evolutionary assumption here is that an old population will accumulate a lot of mutations over time.
Sure.
So they looked at that and they said, oh, they made an inference. That quote there, we infer that Africa is the likely source of the human mitochondrial gene pool. Because there are two big branches here, that word ancestor. That word ancestor there. There are two big branches. To the right or to the bottom is one branch that leads only to Africa, and to the top, other African sequences. They assumed that over time, all these mutations accumulated in Africa. And then one branch arose, and that one branch escaped Africa and spread out across the rest of the world.
Okay.
That's where that comes from.
Now, you say mitochondrial Eve. Do they believe when they use that term that it goes back to one woman as well?
They do, but they don't believe she was the only woman alive at the time.
Okay.
This chart, though, has some assumptions behind it. I want to point that out for the audience. The biggest assumption behind most molecular studies in evolution is called the molecular clock. It's the belief that mutations accumulate at the same rate in all populations. But that depends upon several things. It means that the generation time has to be the same amongst all cultures.
That's not true.
It means that they have to have the same number of children. The children have to have the same death rate. The population has to be about equivalent in size. And there can't be any mutations that affect the workings of the DNA repair enzymes. If someone has a defective DNA repair enzyme, they'll accumulate more mutations.
Sure. And
so there can't be any population differences in any of these things. If those things are satisfied, then maybe out of Africa is true. But every single one of those has been questioned, even in the evolutionary literature. So even the evolutionists are arguing about the basic fundamental assumptions behind out of Africa. So I'm quite comfortable here introducing some ideas that lead toward out of Babel instead.
Out of Babel instead of out of Africa. All right.
Okay, some things that we have to know. First of all, mutations accumulate very quickly in small populations. Now here's the most extreme example I could think of. A population of 2 individuals, and every generation are only 2 individuals. So generation after generation after generation, 2 people, all the way through forever. If one of those people pops up with a new mutation, such as this green-faced person. Now he's not completely green because, remember, we have 2 chromosomes. So one of the chromosomes is defective, the other one still works, he's only half green. Well, there's a chance that he's not going to pass on that mutation to his children, and the green mutation will just disappear. But there's another chance that he'll pass the mutation to both of his children. If that happens, well, maybe they can pass 2 copies to each of the children. All of a sudden, the red mutation, the red face, not the non-mutant gene, the red face gene, is gone. So in small populations, you can get rapid changes. And actually, it's usually very bad because of inbreeding. Inbreeding is bad because you get a rapid fixation of new mutations. So what does that mean for Africa? There's a paper that came out in 2008. And in this scientific paper, appeared in the American Journal of Human Genetics, they quoted early human populations evolved separately for 100,000 years. What does that mean? It means that they're talking about Africa here because they believe Africa is the earliest. They're saying that the African population was divided up into small groups of people, and they stayed separate and small for a very long time. Wait a minute. Under those conditions, you would expect them to diverge rapidly. You would expect new mutations to come, the populations themselves to spread out very quickly. That might explain why Africa has so much diversity in it. Africa has more genetic diversity than the rest of the world put together. In fact, all of Europe has about as much genetic diversity as one African tribe.
Wow.
And 2 people living near each other from different cultural groups in Africa can be more different from one another as a European is from someone from China.
Wow.
It's huge. The assumption, though, is that everything across the world has always happened at the same rate, and I question that assumption.
Yeah.
Here's the out of Africa model. As you go away from the Middle East, in any country in the world, any direction across the world, genetic diversity decreases because fewer and fewer people made it the further away we go. So less genes were carried to the extremes, to the point where the most homogeneous populations in the world are amongst Native Americans. But that's also true in Africa. As you travel south in Africa or west in Africa, genetic diversity decreases. The hot spot of diversity is up in here. Man, that's right next to where we think the Tower of Babel is. In fact, it's very difficult to tell the difference between these 2 spots.
So the assumption that it starts in Central Africa is nothing other than that, an assumption. If you just move it a few hundred miles north and east,
yes,
you're really back to the same map that we have for the Tower of Babel.
Yes.
That's amazing. And really, to verify that, as it moves west and south and west and just west, you see the same diversity you see going the other way from there.
The highest diversity in the world of genetics is in this region.
So the only real difference is where you put the pin for the place where it started.
Yeah, and it depends upon your assumptions about what happened in the past.
That you're making. That's fascinating.
So here's the world. This is what the Bible says.
Yeah.
That at the Tower of Babel, God separated the nations according to language. He used language to separate them, and they spread out across the world. And that is exactly what modern genetics has discovered, a single dispersion of people across the face of the planet. Well, there are a whole lot of different creation stories from people all over the world. What's the chance that the ancient Hebrews hit the nail on the head? About 0 if you're going to make up something.
Right.
Yeah.
Unless there's divine revelation.
Unless there's divine revelation. So this is a schematic of evolutionary history of mitochondrial lines. Now, the letters and the labels, they're just names of groups of mitochondrial sequences that the evolutionists have come up with. So I don't question their labels. L0 is the most ancient, that's the prototypical, that's where their mitochondrial Eve is. L0 in Africa, there's some mutations that accumulated and developed line L1 and line L2, and then line L2 mutated a little more and developed L3. And then inside Africa, L3 eventually mutated into 3 lines, M, N, and R. And these escaped Africa and spread out across the rest of the world.
The L's didn't get out of Africa, but the M, N, and R did.
Yes. Okay. These 3 are found on every continent, including Africa.
Yes.
These are only found within Africa.
Okay.
Okay, so that's their model. However, how do we know that's true? How do we know we started at L0? What if, because N, M, and R are found all over the world, what if these are closer to the original source? What if this is closer to Eve? And then some people made it to Africa, and not all Africans, because there are Africans in this group also. But some of the African tribes remain small. And over generations, more mutations accumulated and fixed in that population so that they diverge from one another, to the point where we have this. This is Eurasia and North America. Here are African sequences. Here are African sequences. Look how many mutations have accumulated in these sequences from the beginning. The question is, well, where do we put mitochondrial Eve? They put it here because they believe in a molecular clock. There should be the same number of mutations from here to here as there are from here to here. But that depends upon your population history and so many other things. So here's how we can explain this using the Bible. We have Eve. She's the mother of everybody. Eve has a mitochondrial genome. She passes it on to her children. Her children pass it on to her children. In the creation account we read in Genesis, there are 10 generations between Adam and Noah. Well, the Bible also describes 4 women being on board the ark, Noah's wife and his 3 daughters-in-law. So we can have about 10 generations from Eve to the 3 daughters-in-law. I don't know if M, N, and R are the 3 daughters-in-law, but it's highly, highly suspicious because there are 3 lines and they're found everywhere.
It sure is amazing, and it sure fits. I mean, the fact that lines up so beautifully with Scripture.
It's a beautiful, beautiful alignment. And if we look at N and R, there's only 1 mutation that separates them. Between M and N, there are 8. So possibly between 1 and 8 mutations occurred between Eve and these women who were on board the ark.
And you said to me that the difference with only 1 mutation between N and R makes it likely that they might have been sisters.
They could have been closely related.
So that 2 of Noah's sons may have married 2 sisters.
Yeah, yes. If the flood is 4,500 years ago, average human generation time is 30 years. Well, if that's true, there have only been approximately 150 generations from the flood until today. That makes us all kissing cousins. But also, when we look at these groups today, they differ by about, people today, like if you and I belong to M, we would probably be about 22 mutations different. So in these 150 generations, we've picked up an additional 22 mutations. And between 1 and 8 from Eve to here in those 10 generations. This fits very nicely within known mutation rates. It's high, but it's not extraordinary. And people don't realize, but Eve is known. We know what Eve's sequence was. And here's why. If you take all of the mitochondrial DNA in the world, it's only 16,000 letters. You can work with it on a laptop. It's a small file. You can line them up. And when you line them up, you realize, like, man, 85% of the letters out of those 16,000, everyone in the world has exactly the same letter. That means Eve had that letter.
Okay.
If you look at the places where there's variability, it might be like everyone in the world has an A and one person has a T. Well, what did Eve have?
She had the one.
She had the A, more than likely.
Sure.
And if you apply that logic,
Probabilities are huge, yes.
you realize suddenly that Eve, almost all of Eve, is known easily. Now, there are some African sequences that are very divergent from the rest, but remember, they're only in one group. They're in groups of African sequences that are different. And this kind of information here is what we'd expect to see if the genome is young. Here's a paper that I had published in an evolutionary journal, Nucleic Acids Research. I was studying mitochondrial diversity within modern people, and I recalculated Eve. And I looked at the diversity amongst people. These are just counts of sequences. I said most people differ from one another by about 22 mutations. So if I take Eve and I compare everyone in the world to Eve, most people are within just a few differences from Eve. But there are some Africans that are way out here. Now, that's the question for the evolutionist. Is that an old population? Or is it a population that's accumulated more mutations over time? Now, I don't want to be accused of being racist here.
Right.
Just because I'm saying there are some, not all Africans, because some Africans here, but just because I'm saying there are some Africans that are very different, it doesn't mean they're mutants or they're worthless or they're weaker than anybody else or not as smart as anybody else.
They're just different.
They're just different. Because we can accumulate a lot of mutations, little ones, and it doesn't do anything. Because there's very poor correlation between the number of mutations and fitness. So I'm not trying to say they're way out or highly mutated. And everyone in the world carries mutations. There's no way around it. We're all mutated. If we look at those letters, we line up all these sequences, and we can find those 85% of the letters, everyone has exactly the same letter. That's this bar right here. There's a small fraction of the letters, however, where most people have an A and one person has a T, or everyone has a G and some person has a C. That's this bar here. But if the genome is very, very old, you would expect more and more of these locations to be mutated. And a lot of mutations, maybe position number 500. One person has a G, one person has a C, one person has an A, one person has a T. You'd expect over time this bar to get smaller and these bars to get taller. But they're not.
Which indicates a young gene pool.
Which indicates very strongly a young genome.
Yes. It's amazing.
And we have some beautiful pictures in the world. This is, remember, L3 is the African group, right? They made M, and then M spread out across the world. Well, this is a picture, a starburst pattern from India. People looked at the M group in India, and these are individual people who are alive today. And look how many mutations they've picked up in the several hundred, several thousand years since they've arrived in India. This is evidence that when the people landed in India, the population started growing, and it's been growing ever since. Stable. That gives you a star-like pattern, not a stair-step pattern. Over here, there's a burst, a second burst. There's like a stair-step here, a second burst of people. Well, this is another star-like pattern that occurred when where some people got to a group of islands out in the Indian Ocean called the Andaman Islands. You had another boom from a common ancestor. This is the kind of stuff we expect with one dispersion of people in the past and a young genome. And we find patterns like this all over the world.
Which makes it consistent with the Word of God.
Which makes it consistent with the Bible.
We got some more to say about this, and you aren't going to want to miss it. We have to take a break, but don't you go away. We'll be right back.
Today's guest on Origins, Dr. Robert Carter, has a PhD in marine biology with a focus in genetics. He is currently one of the senior scientists and a speaker for Creation Ministries International USA. Dr. Carter believes that the human genome is an excellent confirmation of biblical history. He continues his groundbreaking genetics research, and you can find many of his published articles at creation.com. To contact Dr. Carter, write to Creation Ministries International USA, Box 350, Powder Springs, Georgia 30127, or call 800-616-1264.
We are back with Dr. Robert Carter. We're talking about Eve, and we need to go back to sub-Saharan Africa. What is that?
Sub-Saharan Africa is a term. It just means anything south of the Sahara Desert.
Right.
The people north of the Sahara Desert are very European and Asian in their genetics. If you think about world history, there's been a lot of migration and interaction. But the people south of the Sahara Desert are very different from everyone else in the world.
Okay.
And we have to be able to address that.
And so address it.
Okay, well, talking specifically about the out of Africa theory, I mentioned earlier there's a lot of assumptions that go behind it. And the first big assumption is the molecular clock. Well, I have listed here 3 different papers that have specifically questioned the molecular clock in Africa. They're saying there are some African branches that are this big and some are this big, and they say the molecular clock doesn't work. Also, there is abundant evidence that natural selection occurs amongst mitochondrial lines. If you think about someone who lives in Siberia, you might get a very strong selective force saying you have to have very efficient mitochondria or you're going to die because it's cold up there. Well, if natural selection occurs, the assumption now under molecular mitochondrial clock is if you've got 2 people and they're separated by maybe 20 mutations, that the ancestor's right in the middle, 10 mutations from either side. But if natural selection's occurring, it drives differences, and this person might accumulate more mutations than the other, and all of a sudden you don't know where the ancestor is. So we can't build a beautiful tree with a root in Africa if any of these things are happening. Now, it's important for us to know, but there's more. If there's a problem with the DNA repair enzyme, a person is going to accumulate more mutations over their life. And we know that there are people like that that exist, and if there's a difference between two populations, either a defective or a different DNA repair system, it's going to drive the population apart. That's because the DNA naturally breaks and gets repaired every day in your body. But also, Bruce Ames, who's a member of the National Academy of Sciences, he's introduced what he called the genetic triage hypothesis. He's saying that DNA mutation is strongly dependent upon environmental causes. So imagine a population that's under starvation or
Yes.
long-term stress. Well, DNA repair is not necessary for survival. Basic metabolism is. He's saying that if you're under long-term stress, parts of your body shut down, and DNA repair is one of those. So under stress, you accumulate more mutations because you got to live till tomorrow. Who cares if you die of cancer when you're 50?
Right.
So your body turns things off that aren't essential. Well, if that's true, oh man, where's the harshest environment on Earth? Africa is the only major population that spans the equator.
Yes.
India is north of the equator, Australia is south of the equator, and then the equator in South America goes up over the Andes Mountains and then down through the Brazilian rainforest. So currently, in historical times, the only large population spanning the equator was Africa. Now, Genesis chapter 10 says something really interesting. It says, "These were the families of the sons of Noah according to their generations, in their nations, and from these the nations were divided on earth after the flood." I believe that that verse right there is actually the key to understanding modern genetics. Here's why. When we look at the variation between the man's Y chromosomes and the female's mitochondria in the world today, we see that the Y chromosomes have very little variation, indicating we came from a common ancestor in recent times. Also, the Y chromosomes are geographically specific. There's African Y chromosomes, British Y chromosomes, Chinese Y chromosomes. Because these are mutations that have occurred in the lines after Babel, because God divided people according to their Y chromosome. The family lines are based on the male ancestry, and He sent them across the world. But if we look at Eve, we look at the mitochondrial DNA, first of all, there's more diversity, because we came from 3 founding mothers who were on board the ark and came off the ark. But second of all, the female lines, those 3, the M, N, and R, they're scrambled and on every continent. Because God separated the nations according to the male ancestry, not the female. The females were mixed in there, and they got mixed up and stayed mixed up as they spread out. And yet, evolutionists are looking at this with a big puzzle. Because the men are the people who move around more often in history. They go off to fight wars, and they're on long trading expeditions. The women tend to stay at home. And yes, the females' lines that are mixed up all over, and the males' lines that are specific to certain locations.
Which is amazing.
It is totally amazing and fun.